has about 3.2 billion letters of code in a single DNA molecule inside one cell of your body. You have approximately 4 trillion cells that make up your body. Ish. You don't need all 4 trillion cells. Almost every cell in your body has DNA in it. And almost every cell in your body has the same DNA in it. So from something like, I used to be able to do this, a hair follicle, or some saliva, or a blood test, you can get DNA out of an individual, of a human. This, in the grand scheme of things, is not a huge amount of data. 3.2 billion bits is not that big. But in order to understand this 3.2 billion bit of data, sequencing technology chops this 3.2 billion letter molecule into segments on a good day that are about 200 bits long. Ish. You get a whole bunch of these. Lots and lots and lots of them. But chopping them up and looking at them in these 200 bits of data at one time seems pretty easy if you could keep them all in linear sequence. But you can't. And so instead, this 200 bit and that 200 bit, you don't know where they go along this 3.2 billion line. Now it complicates things because our modern sequencing technology also creates these 200 bits. But it does it many, many, many times. And so this 200 bit and this 200 bit have some that are in common but some that are not. So in order to get a DNA sequence out of an individual and have some confidence that you've got the right code, you have to sequence every single letter of the code 20 to 30 times. Yeah.
Why only 20 to 30 times?
Because this allows us the mathematical confidence interval to know that this point right here, being sequenced 20 or 30 times, we get enough consensus across here, we can call this an A. Right? That's a lot of data. That's about 250, 300 gigs of data per person every time you sequence them. Our machines that we use here at the Institute can run, Donna, help me here. There's 16, right? There's 16 microfluidics?
Switching over, so I'm not sure. I mean, they've switched to, I think they're down to 10, but the 10 have a higher output.
Right. So somewhere between probably 16 and 32 experiments at one time. That's people generating this much data across this sequencing platform. So we now have the power to sequence thousands of individuals and look at their DNA sequence all at one time, which is awesome. But in order to get to this, you have to understand a little bit about DNA. So let's back up. DNA is this really amazing molecule. DNA is this really cool double helix kind of thing. And whether you want to believe me or not, Watson and Crick published it first. There's a lot of controversy about who discovered it, but Watson and Crick published it first. They elucidated the mathematical structure behind this. And it's this middle part that holds the code. So these are just backbones. And you have these different molecules that exist inside these two rungs. Adenine, thymine, cytosine, and guanine are their names. You don't need to worry too much about that, but A, T, C, and G is how we abbreviate them. And in order to make both sides of this kind of ladder structure, this helix structure, they pair. And they pair consistently, right? They're molecules, so they kind of fit together like a lock and a key. So this big long string of letters is the DNA code. And within this big long DNA code in a human, it's divided out into 23 different segments. So DNA in a human has 23 segments of DNA inside every cell. Now, because you are human and because you have a mom and a dad, you get 2 copies of each of these 23 bits for a total of 46. So in a human, there are 46 bits of DNA, chunks of DNA, and we call these chromosomes. So that becomes important in challenge 1 and 2. All right, chromosomes are important. And knowing information about where things exist on one of these chromosomes is important. And all the chromosomes are different sizes, and they all contain different amounts of genes on the DNA, which we'll get to in just a second. But a chromosome at its basic level is a big long strand of DNA. Make sense so far? All right. So now we're going to dive into one of these chromosomes and into one of these sequences of DNA. And I'm going to simplify things here. And instead of making this double helix like I have up here, I'm going to draw a line. But that's going to represent the DNA molecule. Okay? So we're going to take a look at this DNA molecule from one chromosome. Doesn't matter which one. Let's say it's number 14. It's a section of it. Lots and lots of letters across here. Hundreds of thousands of letters across this strand of DNA from chromosome 14. And within that hundreds of thousands of letters, there are different things, different markers we can find. So that little piece right there is important. And then maybe down here, there's another piece that's really important. Maybe down here, there's another piece that's important. Okay? Anywhere along this code of DNA that a cell uses the sequence of to create a product. So that product could be a protein, it could be an RNA molecule, it could be different molecules in the body. We call that a gene.
In the bed file, what in terms of the length?
The bed files? The bed files are regions of the DNA, right? So it might be this region.
Yeah,
It might be this region.
what in terms of the length of that, what you drew in there? Like, there's some short one versus
Right. Some are shorter, some are longer, right? Some of it depends on their function. Some of it depends on how complicated these products are, right? So the largest gene in the human body is called titin. And titin, Donna?
I would say about 85.
Yeah, 85 kilobits. Yeah, it's 85,000 letters long, right? That's a big one. And then there are really, really short ones that are maybe 4 letters long that are important sequences. 20 is the smallest gene? Oh yeah.
When you say kilobits and just bits in general, what are you referring to?
The letters. So we call it's kilobases. So each letter is called a base, right? And just like in computer code, we have bits, or bytes, kilobytes, megabytes, gigabytes, terabytes. In genomics, we have bases, we have kilobases, we have megabases, right? I don't think we go beyond that usually. We probably don't have gigabases. I mean, I guess you could, but we don't, right? So those are kind of our demarcations. that's the same log scale, right? It's the same base 10 scale. So this is called a gene. We're going to dive into that in a minute. But a gene is a sequence of DNA that leads to the production of a product. That's very basics. There are other important places along this DNA sequence that affect this thing being made. Some of them are what we call upstream, and they can do things like enhance the production of that, so they're called enhancers. Some of them are downstream. So it could be a repressor, things that lower the production of it. But there are all kinds of other elements that exist along this DNA sequence that are important to this thing being made. And what's really fascinating about this DNA molecule is that these other important things that affect the making of this thing could be on the same strand of DNA, or it could be on a completely different one. Because this thing could interact with that thing in a physical way. And those bed files, that's exactly what we're looking for, is how do these other elements interact with places of interest like genes? How do they physically interact? Because there's a physical binding, a chemical binding with proteins and other structures that bring them together. Does that make sense?
So how do we predict this on just computationally?
So we don't actually predict it computationally generally. There is a technique that we do in an experimental lab that freezes these things bound together. And then when we run that experiment, we can physically see where they bind. Yeah, so it's not necessarily a predictive computational. This information of where that happens is, those are those regions in the bed file. This region is binding with that thing, and we need to pay attention to something in this region. Does that make sense? Yeah.
How accurate do those experimental results?
Well, see, that's part of the problem, right? Is that if you run the same experiment, let's say we're looking and we find this thing interacts with that thing, and we run it today. And then 2 hours later, we're going to run it again, exactly the same way, with the same protocol, the same ingredients, everything. We might find that it's not this segment, but it's that segment. It might be this segment next time. That's part of the challenge. And so it's only by finding the consistency across multiple experiments, looking at the same data, that we can find our confidence that, this must be the region.
How long does it take for you to confirm the result?
So we can run these on the same kind of sequencing platform that we have, and you can run hundreds and hundreds and hundreds of these experiments in a day. So you can generate lists of hundreds of thousands of sequences hundreds of times in a single day. Yes, sir?
Is there any lab in the world automating that to be thousands a day?
There's lots of people working on all kinds of those experiments. There's a timing that has to happen with the chemical reactions that you have to wait for. And so that's really the limiting factor. All right, so basic structure of DNA, 4 letters long, in lots of different orders, lots of long sequences. You have these things called genes that lead to a product. You have lots of places along the DNA sequence that interact with those. And there's lots and lots of genes. In the human DNA sequence, that 3.2 billion, there are about 22,000 regions across that 3.2 billion that we consider genes. Right? And there are hundreds of thousands of all these other things that are interacting, causing things to come on, turn off, go faster, go slower, all those kind of pieces. All right, so that's your basic structural stuff. Now the real question is, why do we care? So I'm going to use the example that you're actually using in challenge 1, right? Huntington's disease is a neurological disease that affects individuals in adulthood. Typically, early onset is somewhere in the 30s. Late onset can be as late as 60 or 70. But it is a neurological wasting disease, which basically means that the body's nervous system begins to shut down. It stops functioning. And so individuals with Huntington's disease lose their motor ability first. They lose the ability to move their hands, their feet, their arms, their legs, eventually their entire body. They lost control completely of it. Eventually, it affects their central nervous system as well. And so things like breathing become very, very difficult. Eventually, all the automated kinds of smooth muscle tissue that you have, digestion, for instance, that stops functioning. But your mind stays. So you are cognizant for much of this. It is a terrible disease, and it is caused by 3 letters of DNA in the code that repeat too many times. Everybody has this set of 3 letters, and they repeat so many times in most humans. But if those repeats go too far, there's too many of them, you get a disease called Huntington's disease. That's the gene region. But there are lots of places upstream and downstream that interact with that, just like all genes. And by understanding how these upstream and downstream things interact with it, we're probably never going to be able to get rid of the repeats. But we might be able to develop therapies that interact on these other places in the genome that interact with that reduce the symptoms, or prolong life, or reduce the onset of symptoms. So we're not trying to find all this information to figure out how to change the genetic code itself. We're trying to figure out how do we understand how this piece of genetic code is being interacted upon by all these other things, and can we use those for targets to help treat this disease? There is no cure, and there's almost no treatment for Huntington right now. So if we could figure out all these interactions, we might be able to find one. So we care about all this stuff. We care about the DNA molecule. We care about the letters. We care about the enhancers and the repressors and the places that interact with those genes and how they function and the code itself, because it leads to things like better understanding of how disease progresses. And it leads to things about how we can understand human nature itself. All right, there is your crash course in DNA and genetics. So who's ready to go be a geneticist, right? Yes. All right, let me introduce our next speaker.